My thesis advisor is Professor Aniruddha Datta, who is a graduate director at the Department of Electrical and Computer Engineering as well. And this project was done in collaboration with our partners at the Department of Soil and Crop Sciences, specifically Professor Endang Septimigish and her PhD student, Ms. Khushbu Rastogi. So this, the research problem that we worked on here was brought to our attention by them, our collaborators. And specifically what we were interested in finding in this paper was the genetic regulators of lysine in rice. And our hypothesis and the long-term goal is that if we identify these regulators, we can go and intervene at them at the genetic level and perhaps increase the lysine content in rice. Okay, so now the question is why lysine and why rice? So lysine is an amino acid. Amino acids are, as you all know, the building blocks of protein, and you need proteins to maintain the proper functioning of the human body. Now, lysine is classified as an essential amino acid as opposed to non-essential because it cannot be synthesized by the human body. So you need to take it in the form of food, specifically proteins. So when we talk about proteins, we think about animal proteins, which constitute about like meat and dairy. Those are like about 28% of the total global protein supply. And in general, animal-based proteins are in demand. That's because they're rich in all these essential amino acid, and the population is growing, which is also creating demands and more pressure on the livestock sector. Now, there are some problems associated with animal-based proteins. One of them is if you are not having the right kind of proteins, for example, if you're eating more red meats or processed meat, you are more susceptible to type 2 diabetes, cardiovascular diseases, and cancers. And of course, the livestock industry has a huge impact on the environment around us. They are responsible for about 14.5% of the greenhouse gases. And the growing demand is only going to exacerbate this matter moving into the future. A viable alternative to this is plant-based proteins, which account for about 57% of the global protein supply. Now, plants aren't as good as animals in their content of the essential amino acids. They're not as complete, okay? So now the thing is, if we can eat a very diverse range of plants in our diet, then it can probably supplement the amino acid requirement. So with that in mind, we focused on rice because rice is a staple of more than 50% of the global population. About 95% of rice is grown in developing countries, and these countries are mostly in Asia. So like every other cereal crops, in rice, the amount of lysine is the least compared to all other amino acids. So we focused on lysine, and we thought that it's a logical step towards enriching lysine in rice and making rice more nutritionally valuable for people around the world. So we dug more deeper into this problem. We looked into the lysine pathways that are implicated in rice. So these are the metabolic pathways, specifically the synthesis and the catabolism pathway. So lysine is produced in the aspartate pathway, along with 3 other major amino acids, specifically isoleucine, methionine, and threonine. And we say that this pathway is self-regulating. That's because when the amount of lysine in this pathway is large, what it does, it uses a feedback inhibition loop to control or suppress 2 key enzymes in this pathway. They are aspartate kinase and dehydropicolinate synthase. So by suppressing them, it effectively stops the production of lysine in the synthesis pathway. Now, in addition to this, lysine can be degraded through the catabolism pathway with the enzymes LKR and SDH. So from an engineering standpoint, what we can see is there are two strategies of maintaining or enriching lysine. One of them is by increasing or enhancing the production of lysine in the synthesis pathway. And the other one is preventing its degradation. Now, this degradation pathway has been extensively studied, and the LKR and SDH genes have been manipulated in rice. And so we focused more towards the synthesis pathway in this paper. And we were interested in finding the genes that controlled the lysine regulation in the synthesis pathway. And the idea is by regulating them or genetically intervening them using technologies like CRISPR, we can effectively increase the lysine content in rice. So to that end, we looked into the genetic regulatory networks that encoded the lysine synthesis pathway. So we derived and constructed this network from the KEGG database. The KEGG database is a public database, so anyone can access it. So each rectangle that you see here is a gene, and they are annotated by their Michigan State IDs. And we have also called them genes A, B, C, D through N because they are very difficult to talk about by their IDs. Some of the different genes here have names. For example, Lys A, DAPF, they are named. So those are a little easier to follow. So we are specifically interested in Lys A because it is a reporter gene of lysine. So what that means is if Lys A is overexpressed or is highly activated, it can lead to more lysine accumulation. So what we are interested in is looking at this entire system and finding out the genes that help in overexpressing Lys A. So essentially what we want to do is we want to go into the system and intervene at one of these genes, A through M, and increase the expression levels of Lys A. So we do this using a technique called Bayesian Networks, and we study the gene regulatory network under both normal conditions and saline stress conditions. We took saline stress here because it is one of the largest cause of crop loss in rice. We have the other big cause of crop loss in rice is drought, and we worked with drought previously in our lab, and we have some papers published for that. So we decided to change our focus in this case. And now I'm going to talk about the Bayesian network and the model and how you can use it in any other gene regulatory networks that you might be working on. So a Bayesian network is basically a data structure that encapsulates or represents causal and probabilistic relationships that are happening in gene regulatory networks. It has the ability to integrate both pathway information, that is the connection between genes, and experimental data. So you can use RNA-seq or microarray data that you might have. So it's a very good paradigm for modeling genomic pathways. So let's take an example. So suppose you have a gene regulatory network where you have 4 genes, A, B, C, and D. And let's say your gene A controls B and C, and D is being controlled by B and C. So we call this structure that you see here a directed acyclic graph. And each of the genes in the graph is called nodes. These are just the technical terminology that you will see in the literature. And the arrows, so the connections need to be pointed in some direction, and those arrows are called edges. Now, for the sake of the mathematics that's to follow, we are going to assume that each of these genes are either turned on or turned off. So they're either active or inactive. So for example, if gene A is active, it may turn on gene B and gene C, or it can turn them off or do one way or the other. So let's just think of it as a binary system where the genes can be either turned on or turned off. With that in mind, so now you have a structure. So you know the biological structure of the genetic system. Now, here's the part where you integrate your data, and this is where the math comes in. So each of the node, right, or the genes that you have they have a parameter. So every model has a model parameter. So these genes have a parameter which are probabilistic in nature. So node A has a parameter called P of A, which is the probability of node A being active. So node B has a parameter called probability of B given A. That's a conditional probability because it depends on the status of A. And similarly, if you look at node D, it depends on two nodes, B and C. So it depends on the status of those two, and the probability of D given C is basically the activation of the gene D conditioned on its parent genes. So in a probabilistic system like this, what you're typically interested in is the joint probability, which is the probability of A, B, C, and D. That's the last expression on the page. So this is usually pretty difficult to calculate in general scenario where you don't have a Bayesian network. What the Bayesian network does is it allows you to decompose this joint probability in terms of the network parameters. And that makes the computational complexity of the model less expensive. And by knowing the joint probability of a probabilistic network, you typically know everything about that system. So that's why we are so interested in it. So all that we have to do is find out what are these values, what are these parameters, probability of A, probability of B given A, how do we estimate them? So we estimated them using Bayesian estimation technique. So if you are estimating a random variable X with some data set D, you in a Bayesian framework, what you do is you don't come up with a number, but instead you come up with a distribution. So you come up with a range of values for X, and each of those values have some probability of occurrence. And those two things are determined by the likelihood, that is the data, and the prior distribution on X. So prior distribution means what is your initial assumption of what X should be. So as you have more data, the posterior distribution converges to the actual value of the probability. So if you have a Bayesian network where you have a node X and you want to find out its probability of activation theta X using the Bayesian framework, and you assume that you're working in the binary domain. So you have some data, binary data with n observations, and let's say about k of them are ones, then the likelihood is following a binomial distribution. Now you have the likelihood, you have to choose a prior distribution on theta X. Now the choice of theta, the prior distribution on theta X can be designed based on your domain knowledge, so your understanding of the biology or the genetics, or you can use something that is computationally effective. So for this example, we are using a beta prior, or you can use something like a non-informative prior, which doesn't influence much of your posterior distribution. So in that case, the posterior distribution is driven only by the data. So in this case, we used a beta distribution because it makes the computing of the posterior distribution easy, just for this example. So if you have a beta prior and a binomial likelihood, the posterior distribution also follows a beta distribution with updated parameters. And so essentially the problem is sometimes when you have your own custom prior distributions, you don't find a closed form solution like this, and you often have to use sampling techniques to arrive at an approximate answer. So this is the posterior distribution of the probability, so that you were trying to find out, or the network parameter, but this is a distribution, it's not a number. And in order to get a number, what you have to do is you have to approximate the distribution to its expected value or the average value. And that's usually a very standard approximation and in the Bayesian framework for getting a point value. So now the thing is, you can use this Bayesian estimation to find out all these values, P of A, P of B given A, C given A, and this one, D conditioned on B and C. So now that you know this, your Bayesian network is completed. Now all you have to do is you have to run this Bayesian network. So how do you run it? So let's say we go and intervene at gene A. Let's say we turn it on. And now we want to see what's the effect of turning this on gene D, okay? So we can write the conditional probability using Bayes' rule as the ratio of the joint distribution divided by the marginal probability of A. We can express the joint distribution in terms of the whole system by summing out variables B and C like this. And then we already saw in the previous slide that the joint distribution can be written as a product of the network parameters. And we can cancel out P of A from the numerator and denominator, and we get this. And this is a sum product equation. And each of these terms that you see in the equation, they are network parameters that you already estimated from your data. So you can essentially calculate this and be done with this calculation, and you can actually find out what is the effect of activating A on the gene D. So that's pretty neat. And this technique is called the exact inference algorithm, where you calculate the probabilities accurately. Now, these techniques are usually very good for simple networks and which follow a certain structure, such as trees and polytrees. But when you have complicated structures with loops, it becomes more difficult to use these algorithms and these exact inference algorithms. So in our paper, we used a more sampling-based approximate inference technique called likelihood weighting. It's based on Markov chain Monte Carlo. So what you do is you have the network parameters, and those are your distributions, and you draw samples from them. And the idea is the more number of samples you draw, your probability will converge to the actual values. So we used that in our computation. Now I'll talk about the experimental setup and the results that we got. So as you know, like the Bayesian network structure was derived from the KEGG database. We used an RNA-seq data which contained rice under both saline stress and normal stress conditions. It contained rice seedlings under such conditions. And there were 368 samples per gene. We normalized the data using the median of ratios technique. That's the same technique that you used for in the R package DESeq2 for differential expression, gene expression analysis. Then we segregated the data by the normal condition and the saline stress conditions. We quantized the data into 3 segments: inhibition, dormancy, and activation states. So in the example, we looked into binary states where you had activation and inhibition. Here we are looking at 3 different states. We used a non-informative prior. It's called the Dirichlet prior because we didn't have prior information about how each of the genetic nodes are, what their states should initially be. So we only wanted the data to drive our conclusions about the network parameter. Then we ran our interventions on single nodes, and then we performed the intervention in pairwise fashion. And we computed a score where we measured the probability of activation of lyse A, and we subtracted the probability of lyse A being active without any gene intervention. So the higher and the more positive that number is, the more effective that gene intervention strategy is. So for single node intervention, that's when you went in and turned on one gene at a time, like we saw in the example. So we see that under both stressed and unstressed conditions, gene M, which corresponds to DAPF, when it is activated, it turns on, it has the highest score. What that means is when DAPF is being activated, it has the highest potential for increasing the lysase activation. We also see there are midstream genes like I, J, K, and L, that are implicated in the single intervention strategy. You can see as the stress is between stress and unstressed conditions, that gene L is active, and in stressed condition, gene L needs to be dormant. So basically the idea here is that gene M or DAPF has the best potential of activating lyse A. We also checked pairwise intervention, where we went and intervened at genes 2 at a time. And we see more upstream genes like A and C being activated. But what we noticed is that these were always paired with the gene M or DAPF, and DAPF was always activated. This we observed under the normal conditions and also under saline stress conditions. So that was pretty significant, and it bolstered our understanding that DAPF might be a good gene that we can go and intervene at. So for our future research work, obviously these are simulation results, and we will need to validate them experimentally in the field. We have had success with this in Arabidopsis in modeling its drought signaling networks. So we have some good hope for this. We want to also expand our system boundary and include the amino acid threonine, and because it affects the aspartate kinase, which is in the lysine synthesis pathway. And as and more literature becomes available, we would like to update our prior distribution from Dirichlet to more informative priors, so that our estimation of the network parameter becomes better. And with that, I'll conclude my talk for today. And you can contact me at my email here, and you are free to ask me any questions. And I thank the organizers for having me here today. So.